If everyone would please settle down, the webinar is about to start soon. Before we begin, I would like to ask if I am currently audible. Mic check, mic check. Could our participants please give a thumbs up if you could hear me? Okay, thank you. Please be advised that we will be starting our webinar in 2 minutes. Let us all rise for the singing of the Philippine National Anthem.
Bayang magiliw, perlas ng silanganan, hanap ng puso, sa dibdib mo'y buhay.
Lupang hihirang, duyan ka ng magiting. Sa manlulupi, di ka masisingil.
Sa dagat ang tulong, sa simoy at sa langit mong lugaw, may dilag ang tula at awit sa paglayang minamahal.
Ang iskap ng watawat mo'y tagumpay na nagdidingding.
Ang bituin at araw yakay ka,
May di magdidiin.
mamatay ng dahil sa'yo.
A pleasant day to everyone. Good morning. Wherever you are in the Philippines or maybe in other parts of the world, welcome to HugNayan Online, Search for Life Beyond Earth. I am Robert Talabis and I'll be your host for today. It's fantastic to see such a strong turnout today. Thank you all so much for your interest, time, and effort in joining us in this webinar. I see that our participants are hailing not only from different places, but also from different universities and institutions. Before we formally start, I'd like to ask the audience to comment in our chat section their institution and where they currently are. For example, I am Robert Patrick G. Pavis from the Philippine Space Agency in Quezon City. Through our comment section for our participants. We have here Miss Elise Angela Gamana from the Philippine Space Agency, also in Quezon City. Okay. So great to see our audience from all over. We have Miss Althea Reyes from Bulacan, Dr. Ernest Macalalad from the Philippine Space Agency. We have Mr. Ronald Dimu from Xavier University at Dineo de Cagayan. Dan from Mindanao State University, IIT. So it's great to see our audience from all over. We have an incredible lineup of speakers ahead, coming from all the way from Europe and the United States. And in line with our webinar today, they come from reputable institutions specializing in astrobiology. We are truly honored to have them in the webinar despite the huge time differences. So do make sure to express our gratitude. To start, the HugNayan Space Time Fora was started by the Philippine Space Agency's Interdisciplinary Space Missions Development Division to expand the knowledge of PhilSA personnel in the field of space science. In this special edition of HugNayan, it is open to a wider audience to further share the wonders of space and beyond, specifically providing foundational knowledge in astrobiology and highlighting recent global research development. With this, it is our greatest pleasure to introduce our opening speaker to set the tone for our session today. Please give her a warm round of virtual applause, the Ad Interim Director General of the Philippine Space Agency, Dr. Gay Jane Perez.
To our distinguished speakers from the University of Naples, Federico, Miss Paola Manini, and Miss Savannah Zigic, and to Mr. Bernard Ayala from University of Florida, colleagues from the Philippine Space Agency, Dr. Mark Cesar Talampaz, Dr. Rainel Reyes, Dr. Ernest Macalalad, and team, ladies and gentlemen, good morning. On behalf of the Philippine Space Agency, I warmly welcome you all to this year's second installment of HugNayan. Today, we gather to explore a fascinating and deeply thought provoking field, astrobiology, a discipline that asks some of humanity's most fundamental questions, like how did life begin? And are we alone in the universe? This session is one of the first astrobiology focused seminars in the Philippines. It reflects our growing effort to expand national conversation on space science into more interdisciplinary and emerging areas of discovery, areas that challenge us to ask bigger questions and imagine new possibilities. And this conversation comes at a timely moment. Just recently, we witnessed a major milestone in exploration with the successful Artemis 2 mission, bringing humanity once again closer to the moon and eventually deeper into space. We push further outward and we are reminded of something equally important. Even as we look on our planet, our understanding of life and our responsibility remains grounded here. At the same time, popular culture continues to spark curiosity and imagination. The recent Project Hail Mary has captured public interest in interstellar travel and in the search for life beyond. Stories like these often begin with a simple but powerful question: what if? That question matters because science is a continuous process of discovery. It begins with curiosity, is sustained by persistence, and grows through our willingness to explore the unknown. I encourage you to hold on to that sense of curiosity because it is often the starting point of something bigger. I would also like to take this opportunity to thank our distinguished speakers for joining us and for sharing their knowledge and expertise. We are especially proud to highlight Ms. Angeline Macasinag, a graduate of PhilSA's Advanced for Accelerating Space Science Research and Development and Applications, or AD-ASTRA, scholarship program, who now contributes to our work here at PhilSA. Her journey reflects how Filipino talent can be nurtured and empowered to take part in advancing space science and research. Together with ours from the international community, your presence reflects the collaborative and global nature of this field, where knowledge is shared across borders and discovered a collective effort. As we begin, we are reminded that the search for life beyond Earth is more than a scientific pursuit, it is our shared journey. It challenges us to rethink what life is, where it can exist, and how we understand our place in space. At the same time, it brings us back to another important question: what do we choose to do with what we know? Knowledge in itself is only the beginning. What matters is how we use it, how we apply it, we share it, and how it contributes to something beyond ourselves. To our students, young scientists, and curious minds joining us today, we hope this more than inform. We hope it sparks questions, fuel imagination, and inspires you to continue exploring not just what is out there, but what you can do with what you learn. And just as we have seen through programs like our Ad Astra Scholarship, the path from curiosity to contribution is one that is very much within your reach. With that, I wish everyone an insightful and engaging learning. Thank you and good morning.
Thank you so much, Dr. Perez, for that wonderful and inspiring opening. I'm sure everyone here has a shared sense of wonder as we dive into the mystery of life and the cosmos. Before diving into our discussion proper, please give me the opportunity to discuss the flow of our program. From 9:25 to 10:15 a.m., we'll have our first lecture titled Astrobiology 101. After this, we'll have a short 10-minute health break. From 10:25 until 11:25 a.m., we'll be having our next three speakers discuss the following lectures: the search for life on Mars, the Earth's cryosphere and search for life on the icy moons, and lastly, methods to detect signs of life. Once all these incredible presentations have ended, we'll be opening the floor to a question and answer session. This is a reminder that you may ask a question through our Q&A box found at the upper section of Microsoft Teams. These will be answered in our designated session. Then we'll have our closing remarks from 11:50 a.m. to 12 noon. Lastly, we are asking everyone to please stick around until the end of our webinar for a photo opportunity. But before we start with our lecture sessions, we have a couple of rules we'd like to outline before we start. First, use your full name in the MS Teams display name. Second, keep your microphone muted. Third, use the Q&A box for questions. Fourth, accomplish the evaluation form. At the very end of our program, we will ask you to accomplish the evaluation form. The QR code and the shortened link to access it will be flashed before we conclude the seminar. For those who would want to receive an official certificate of attendance, an accomplished evaluation form will be required for you to get it. So please stay with us until the very end of the program. And last but not least, observe proper decorum and be respectful. Without further ado, may I call on the Supervising Science Research Specialist of the Interdisciplinary Space Missions Development Division of the Philippine Space Agency, Dr. Reynel Inhosa, to introduce our first speaker.
Thank you, Robert. So good morning, everyone, and to our distinguished speakers and participants. Um, for our first speaker is a fellow crewmate here at the Philippine Space Agency, Miss Angeline Macasinag, who works as a project technical specialist at the Interdisciplinary Space Missions Development Division in PhilSA. So she currently spearheads the research and development effort of our division related to astrobiology. She also currently serves as a board member of the Philippine Space Biosciences Society Incorporated, where she actively promotes research and capacity building in astrobiology across the Philippines. Ms. Macasinag earned her master's in biology of extreme environments astrobiology curriculum from the University of Naples Federico II in Italy through the Philippine Space Agency's Ad Astra Scholarship Program, where she graduated cum laude. She also holds a Bachelor of Science in Biology from Bulacan State University under the DOST Science Education Institute Scholar and also earned a graduate diploma in astronomy from Rizal Technological University. Before pursuing her graduate studies, Ms. Macasinag managed national health R&D projects under the DOST Philippine Council for Health Research and Development for 5 years and also worked as a laboratory analyst in a pharmaceutical industry. Her research interests focus on the survivability, biological response of pigment-producing bacteria under simulated Martian conditions and its implications in Mars habitability, biosignatures, and the survival of microorganisms in extreme environments. So our first speaker, or for our first presentation, let's all welcome Ms. Angeline Macasinag.
Thank you for that, Dr. Inahosa, my supervisor here in PhilSA. So just please allow me to set up my share screen here. Oops, sorry. Okay, sorry, we're having just a very minor technical problem here.
Please hold as we are facing technical difficulties on our end. We will appreciate your patience as we try to start our first presentation.
Okay, I'm really sorry about that. Okay, so now let me start. Okay, is my screen, can you see my screen? Let me see some thumbs up if you are seeing my screen now. Okay, thank you, thank you. Okay, so good morning, everyone, again. And welcome to the first lecture of this webinar with the theme, The Search for Life Beyond Earth. I think it is only fitting to begin with the big question, is there life beyond Earth? And to be honest, I'll give you a little spoiler right away because we still do not know the answer yet. But the search is very much ongoing, and that is exactly what makes this topic so exciting. We have a series of fascinating talks ahead of us today, and my role is in this opening lecture is to provide some introductory context and foundational knowledge about the science behind our search for life beyond our planet. But before we begin, I'd like to help set the expectations, not only for my talk, but for the webinar as a whole. So most of our discussion today, let's say around 99%, will focus on life as we know it on Earth. And of course, that number 99% is just a playful estimate. Our panel of speakers comes from different fields such as biology, chemistry, and ecology. So our conversations will naturally be grounded in established scientific knowledge and in what we understand from life on our own planet. Now, I know that whenever the topic life beyond Earth comes, so many people immediately think of aliens, mysteries, and all sorts of unusual questions. That curiosity is completely understandable. However, in today's webinar is intended to focus on ideas that can be explored through science and evidence. So this Also means we are not going to be discussing conspiracy claims or topics that currently lack scientific support. Those may be conversations for other spaces or platforms, but uh, for today, they are outside the scope of today's session. So as early as now, we would like to kindly ask, please, for everyone to keep your questions later aligned with the topics of our speakers and also with the theme of our webinar. And as for the remaining 1%, we may be briefly touch on the possibilities of life as we do not know it. So speculative ideas that stretch our imagination while still connecting to science. And with that, let us begin our journey here on our little blue planet called the Earth. So our audience today comes from many different backgrounds. So let's begin with something simple and exciting. What does life need in order to exist? So whether we are talking about a human being, a tree or bacterium, or even our cute little dog here, Hachi, every living thing is made up of tiny units called cells. And cells are the basic building blocks of life. And if we zoom in even further, cells are made of molecules, and molecules are made of atoms, the fundamental pieces of matter. So, in a way, life is built from chemistry. Life as we know it on Earth depends on on a small set of key chemical elements. First, there is water. A large portion of our cells in our body is made up of water, and water is essential for nearly every biological processes. So, water is essentially H2O. So it's made up of hydrogen and oxygen. So let's list down here on the left the chemical key elements. So the first one is hydrogen and oxygen. Then we have carbon. So one of the most important elements in biology is carbon, and it is very remarkable because it can connect with many other atoms. With many other atoms and build complex molecules. That is why carbon forms the backbone of proteins, fats, and sugars, and even more importantly, also genetic materials. So life also depends on several important elements such as nitrogen, which is used in proteins and DNA. Another is phosphorus, important also for DNA and RNA, and especially energy transfer in the cell. And another key element is sulfur, which is found in some proteins and enzymes. So together, these six major elements are often remembered as SHNOPs. So remember this acronym: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. So if you're going to have a key takeaway as an acronym today, let it be SHNOPs. So from our biology here on Earth, let us now look upward. So here, when we look upward, here is where things become truly fascinating. So the same elements that make up our body, the carbon, hydrogen, oxygen, nitrogen, and the rest are not unique to Earth. In fact, they are also found in stars, planets, moons, comets, and asteroids. So that means the ingredients of life are scattered throughout the universe. So the carbon in your cells was forged in ancient stars. So in a very real sense, we are made up of stardust. So beyond elements, at the molecular level, it even gets more exciting. So scientists have discovered organic molecules on Mars. So in science, organic molecules are carbon-based compounds, and many are important in biology. So for example, NASA's Perseverance rover on Mars has detected organic molecules in Martian rocks. Now, to be clear, this is not the proof of life on Mars, but it tells us that some of life's chemical ingredients can exist there. And Mars is not the only place. Samples are returned from the asteroid Bennu has revealed a variety of carbon-containing compounds, including molecules linked to prebiotic. So imagine an asteroid traveling through space, carrying some of the same kinds of molecules connected to life. So you might be more familiar with DNA, but we also have other equally important biological material called RNA. So here are their differences. So the DNA contains In terms of function, the DNA contains the long-term instructions for life, and for RNA, it helps carry out many of those instructions. So in terms of structure as well, DNA is generally double-stranded, and RNA is commonly single-stranded. And they are, in terms of sugar backbone, they are different in a way that RNA is built from a sugar called ribose, and DNA is built from a sugar called deoxyribose. In terms of nucleobases also, are almost the same. They are composed of adenine, cytosine, guanine, both, but they differ in terms of DNA contains thymine, while RNA contains uracil. So the molecules needed to build this RNA is found in asteroid belt. So the nucleobases, as I've said, the GCAU, guanine, cytosine, adenine, and uracil, are found in asteroid. And even the sugar, the sugar needed to build the RNA, as I've mentioned, the ribose, is also found in asteroid belt. Also, there is another reason why scientists take this question seriously. Not only that we see similar chemical ingredients or processes in space, we also observe the same physical laws. Say, for example, the force that makes your pen fall to the ground is also the same force that keeps the moon orbiting. And that force is called gravity, and we are all governed by the universal law of gravitation. So perhaps instead of asking, is there life beyond Earth? Maybe a deeper and more scientific question might be, is biology universal? So if the ingredients of life are common, if the laws of nature are the same, could life also emerge somewhere? And once again, I put a little spoiler here. We still do not know yet. But for scientists, that uncertainty is not frustrating. Instead, it is an invitation to keep on exploring. So as I mentioned earlier, this question is not just science fiction or imagination. There is a real scientific field that studies these kind of questions very seriously. And that field is called astrobiology. Astrobiology studies the origin, evolution, distribution, and future of life in the universe. And personally, I admit that I am biased. I think astrobiology is one of the bravest sciences because it asks some of humanity's biggest question: how did life begin and evolve? Um, does life exist elsewhere in the universe? And what is the future of life here on Earth? What also makes astrobiology exciting is that it is highly interdisciplinary. So it is not reserved for astrobiologists. To explore this kind of questions, we need many fields working together. We need, of course, biology to understand life. We need chemistry to understand molecules. We need also geology to understand the planets and environments. We also need ecology or environmental science to understand the living systems, physics and astronomy to understand the worlds beyond Earth. So, in a sense, no single science can answer these brave questions alone. And before we ask whether life exists on other planets, we should ask, how did life begin on Earth? Because if we want to search for life elsewhere, we must first understand how life may have started in the one place we know we exist, and that is Earth. So let us now look at some major scientific hypotheses on the origin of life on our planet. So earlier, I mentioned the RNA, one of the important molecules used by life today. So there is a major scientific idea called the RNA world hypothesis, which proposes that life may have begun in a world where RNA or similar molecule came first. But why RNA? RNA is very special because it can do two very important works. So the first one is to store information like a set of instructions, and second function is helping chemical reactions happen like a simple catalyst. So these are the two key functions of RNA. And The idea of the RNA world hypothesis is that on the early Earth, perhaps in a hot spring, tidal pools, or other active environments, natural chemistry may have produced simple RNA-like molecules. So some of these molecules may have developed the ability to make copies of themselves. And once that happened, they could begin to change, compete, and evolve over time. So as they become more varied, some may have started working together, forming early genetic systems, helping build more chemical proteins, and eventually they created membrane-like barriers that separated them and protected them from the outside environment. And then over long periods of time, these simple systems may have eventually become the first true cells. Another hypothesis that looks at an even earlier stage, before even the RNA and before the first genetic molecules, is called the PAH world hypothesis. So PAH stands for polycyclic aromatic hydrocarbons. So while the name sounds very complicated, these are just simple carbon-based molecules made of connected ring-like shapes, just like this, and they can form naturally on Earth. And surprisingly, they are also detected very abundantly in the universe. So this hypothesis suggests that molecules like PAH may have helped organize the chemical reactions that eventually led to life. So in other words, before cells and genetics existed, simpler molecules may have helped prepare the conditions for life to begin. And for this topic, we are especially fortunate today. We are honored to have a scientist with expertise in this field. Later, Professor Paola Manini will guide us further into the science of PAH and their possible role in the origin of life or our search for life on Mars. Third is another influential idea about the origin of life comes from Charles Darwin himself in his letter to his friend in 1871. And it reads, he wrote to Joseph Hooker, but if, oh, what a big what if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, energy sources like light, heat, and electricity present, that a protein compound was chemically formed and then eventually become complex and then become what we know of life we know today. So he was imagining a small body of water on the early Earth, much like similar to the video on the right, where simple inorganic substances could gradually combine into more complex organic molecules. So energy sources like sunlight or lightning could have driven these chemical reactions. And since this hypothesis is easy to imagine, what other ways to test it but through scientific investigation? So more than 80 years after Darwin wrote that letter to his friend Stanley Miller, and under the supervision of Harold Urey, tested the core idea of the warm little pond hypothesis and used this kind of experimental setup that you are seeing on the right. So they replicated the atmospheric composition of the Earth as described in the warm little pond, such as the atmospheric composition, such as the methane, ammonia, water vapor, which by the way comes from here, and hydrogen gas. So they also imitated lightning through electrodes, and all of these organic compounds reacted. The chamber where the gases were reacting was connected to a condenser, where the condensed resulting products were collected in a collection trap. So after 24 hours of observation, this is what Miller-Urey experiment looked like. So they analyzed what was there in the collection trap, and to their surprise, they see that from the inorganic starting materials, the methane, ammonia, water vapor, and hydrogen gas, they ended up collecting organic compounds, and more notably, amino acids that, as we know, as we know, are important composition of building blocks of life. So this experiment has become one of the most famous experiments in science today. So this experiment became a landmark in the origin of life research because it showed that life's basic chemical ingredients can form from simple starting materials under the possible early Earth conditions. So over time, many scientists repeated and extended this type of experiments. So some ran them longer, producing an even wider variety of organic compounds. A specific group of scientists led by Criado Reyes noticed that the Miller-Urey experiments and even the experiments that repeated them used the same kind of glass composed of borosilicate. So from their knowledge, borosilicate is chemically reactive, especially when conditions are alkalinic and under uh, high temperatures. So this is the very same case in the Miller-Urey setup. So under such conditions, they know that small amounts of silicate species can potentially leach into the reaction mixture and possibly influence the chemical reaction happening. It is also important, so their group asked, what if the container itself is influencing the chemistry? So it is also important to note that silica and silicates are important constituents of rocks. And in order to test the hypothesis, if indeed the glass itself is influencing the reaction, they uh, compare different setups. So the first setup is the standard borosilicate glass. Uh, the one that is commonly used in the Miller-Urey experiment. And the next setup is they use Teflon. Teflon is chemically inert, meaning unlike borosilicate composition, they do not react or shed when the temperature is high and when the temperature is alkalinic. And the third setup is they use the same chemically inert Teflon, but this time they use some bits of borosilicate glass in order to see if the borosilicate itself is catalyzing or influencing the chemical reaction. So their results showed something very important. So the glass can actually enhance both the amount and diversity of organic compounds formed. And this may sound like a technical detail, but it changes how we think about early Earth because if minerals, rocks, or surfaces can help chemical reactions happen, then life's origin are not just about what was in the water in the Miller-Urey, but also about what the water interacted with. So a warm little pond on the early Earth would not have been isolated. It would have been in constant contact with rocks, minerals, heat, and energy sources. So another hypothesis is the hydrothermal vent hypothesis. So this hypothesis tells us that instead of starting at the surface of the Earth in a warm little pan that is proposed by Darwin, according to this hydrothermal vent hypothesis, life started far below the ocean, near the cracks in the seafloor called the hydrothermal vents. So hydrothermal vents are formed when seawater seeps into the Earth's crust, becomes heated by underground magma, and then rises back up, carrying dissolved minerals and gases. So this superheated mineral-rich fluid then meets the cold seawater surrounding it. So where hot fluid meets cold water in the ocean, strong chemical gradients are created. And gradients like this can power chemical reactions. Scientists think these vents may have acted like natural chemical laboratories, which continuously supply important gases such as molecular hydrogen, methane, and ammonia. And when these interact with carbon dioxide that is dissolved in the seawater, more complex organic molecules could have been formed. And because the many hydrothermal vents are found deep in the ocean, sunlight cannot reach them. So how could life possibly survive there? They use, instead of photosynthesis, they use chemosynthesis. So it is a process of using chemical energy instead of sunlight to make their food. So instead of relying on photosynthesis like plants do, some microbes use chemicals from the fluid of the vent as their energy source. So even today, an entire ecosystems uh, exist around hydrothermal vents. So they are not supported by sunlight, but they are supported by chemistry. So this will be further discussed by one of our speakers here as she discussed later on our search for life on the isomers. So this idea is important because it also considers how harsh early Earth may have been. So they also considered that the young Earth could have been exposed to intense UV radiation. Also, it is also frequented with volcanic activity, meteoric impacts and overall the surface, according to this hypothesis, is not stable for life to exist. So they proposed that the deep ocean would have shielded life from these harmful factors. Now, all of the hypotheses that I presented, those for the RNA world, pH world, warm little pond, and hydrothermal vents, they all assume that life or the building blocks of it or came from are formed on the Earth. But there is another idea called the panspermia, which suggests that some of the ingredients of life, or possibly even microbial life itself, may have formed elsewhere. Um, and then they were later delivered to Earth by comets, meteorites, or asteroids. So this idea becomes interesting because scientists have indeed found organic molecules in meteorites, asteroids, as I have provided you with an example a while ago. Asteroid Bennu, we found sugars and RNA um, components, and even interstellar clouds. So all the ideas we discussed today are serious scientific hypotheses. They help guide experiments, observations, and guide us to possible new discoveries. But it is important to remember that none of them has yet fully explained non-living chemical how non-living chemistry became living biology. So that remains one of the greatest unanswered questions in science. And perhaps that is what makes this field so exciting. So we do not have all the answers yet, but we are getting better at asking the right questions. From the origin of life, we now begin our understanding of the kind of way of living that we are not very familiar with, and that is life at the extremes. So even as we see Earth as a seemingly gentle, welcoming planet, it also harbors many extreme environments that will make you question, is Earth pro-life or not? And when I say extreme environments, I am pertaining to places to which conditions are harmful to conventional life that we know. It can be volcanic systems, high pressure environments like the deep sea. We will see more examples later. These extreme environments is very important in astrobiology as they can be a good analog in studying life beyond Earth. But before we move forward, attention check. Let us play a very short game, and also for me to just take a moment to sip some water and catch my breath. So the game is called Earth or Not Earth. So the mechanics of this game is very simple. You're just going to tell me if the pictures set of pictures that I'm going to project here on the screen is uh, found on Earth or not on Earth. So for in the interest of time, if your answer is Earth, you can just write letter E. And if your answer is not Earth, you can just write letter NE. So you can use the chat box to participate in our mini game. So for example, let us just have some two pictures, for example. So the answer to this is the first picture is found on Earth. It is in the Yellowstone National Park, found in US. And the next is not on Earth, but on Mars. So it is taken from one of our rovers there, Perseverance, as we can still see the trails that was captured in this picture. So I'm assuming that everyone is ready. So I'm going to project here four pictures, and you're going to tell me if it is found on Earth or not on Earth. E for Earth and NE for not Earth. Please use the chat box, and I will just give you 30 seconds to answer. Okay, thank you for that. Let us now reveal the answer. So the first picture is on Earth. It is found in Australia, in Pilbara. So it is an extreme environment characterized by exposure to intense solar radiation, especially during summer, making it hot and very dry. The red color also that you are seeing there is because the soil is rich in iron. The second picture is not on Earth, but it is somewhere very, very far away. It is found in one of Saturn's moon, Titan. So this world is the only known world besides Earth with stable liquid water on its surface. But instead of liquid water, it is characterized by liquid hydrocarbon lakes with rivers of ethane and methane. The next picture is on Earth. It is an aerial view of the McMurdo Dry Valleys in Antarctica. So it is characterized by cold and dry environment. Our last picture is not on Earth, but on Mars. So it is the Jezero region, where we believe consists of ancient river. That's why we are seeing delta here. We sent our rover Perseverance there to collect samples to understand the geology and also to see if there are signs of life on the red planet. So from our mini game, we saw that some of the environments on Earth can be mistakenly thought of as an environment from another planet and also vice versa. So in biology, these extreme environments are important analogs for research. For example, the Antarctic Plateau holds the world record for the coldest region on Earth. So microbial life in Antarctic subglacial environments helps scientists imagine how life could exist in Europa's ocean. Europa is one of the icy moon of Jupiter. We will see in the lecture after the break more of this. And the next environment is the Atacama Desert, which is found in Chile. It is also a record holder in a sense that it is the driest non-polar environment in the world, and it is with its other environmental characteristics such as exposure to intense UV and Mars-like geology, it is often used as an Earth analog for Mars. So many instruments being tested before being deployed to space or being deployed to the red planet. We test the rover technologies here and other life detection instruments. Our next example for extreme environment is, if you can remember in history, the Chernobyl disaster. It is actually the worst nuclear accident in history. So Chernobyl is powerful analog for space environment research because its exclusion zone offers a real-world laboratory where organisms, ecosystems, and materials are exposed to long-term radiation stress, which are conditions that are very similar to those faced in outer space. Next is somewhere in Hawaii. We also built space analog station in Volcano to test technologies and conditions for our future lunar and Mars missions. But how about in the Philippines? So in the Philippines, we also have a very good variety of extreme environments. Example here is include sulfur vents in Valencia, Siquijor. The picture and the video here are taken by my friend. So this sulfur vent is characterized by hot temperature and highly acidic environment. Another is Mud Spring in Mount Makiling in Los Baños. So I saw that we also have some students and faculty from the UPLB. Shout out to you. So you have an extreme environment there that you can explore for astrobiology. So in the Philippines, there is currently a limited number of studies in extreme environments in the context of astrobiology, even if we have lots of environments that we can use. On Earth, life is really widespread, even in environments with hostile conditions. So organisms that thrive in extreme environments are called extremophiles. In a literal sense, they are the lovers of the extreme. So these organisms are highly specialized in that they evolved to grow only in these extreme conditions. So they have specialized cellular membranes and enzymes adapted to the extreme environments that they live in. So depending on the environment, they are categorized on as what you can read on the left. We have thermophile or hyperthermophile for organisms that who loves to thrive in a very hot environment. And its counterpart in the cold is called psychrophile. We also have uh, extreme pressure loving organisms called piezophile or hyperpiezophile. They're often referred to also as barophile. Um, the next is for the pH. Those who loves low pH acidic environment are called acidophile, and its counterpart on the alkalinic environment is called alkalophile. So as you can see, the names are just very simple to understand. The next is those that who loves environment that uh, has high salt concentration are called halophiles. And the next we have a non-philic ending organism but tolerant ending. They just tolerate the environment, meaning they do not reproduce, they do not metabolize there. They just become sometimes dormant to be able to withstand the kind of environment. And those are called radio-tolerant organisms. And in reality, environments are not just one factor. In reality, environments are composed of combinations of extreme conditions. And conditions that thrive in multiple extreme conditions are called polyextremophiles. So an example here, for those extremophiles who love both high temperature and acidic environment are called thermoacidophile. And its counterpart, those who loves low temperature with high salinity environment are called psychrohalophile. So to help you imagine, here are some of the examples of extremophiles. So we have here the Methanopyrus candleri. It is a world record holder for growth at a maximum of 122 degrees Celsius, which is way, way beyond the temperature of boiling water, and it was isolated in a hydrothermal vent in California. The next, its counterpart in the cold is called Methanogenium frigidum, which can only live when the temperature is below 15 degrees Celsius, and this organism is isolated in Antarctica. The next is Thermococcus piezophilus, which holds the record for growth observed in extreme pressure. In fact, it is isolated from the deepest known hydrothermal vent in the world. The fourth example is Picrophilus torridus. Its genus Picrophilus holds the record for growth at the highest acidity of pH 1. And for alkalinic environment, our example here is Serpentinomonas, which prefers, on the other hand, highly alkalinic environment with pH 11. Next is Halobiforma haloterrestris, which was isolated in an environment somewhere in Egypt that has a high salt concentration. And usually, this kind of environment also has very limited water activity. Now, probably among the examples I have given here, you are most familiar with this little cute animal, tardigrade, as this is a favorite sample to test resilience in space-relevant radiation. And lastly, we have the world's toughest known bacterium, Deinococcus radiodurans, as they can withstand very intense radiation up to 12,000 gray of radiation, which is far exceeding the lethal dose for humans and most other organisms. So that's what's up with life living at the edge. Our next topic will be a very brief introduction to an important term you will hear many times in the next three sessions, which is biosignatures. So it is very helpful that we understand it now. A biosignature is any substance, molecule, pattern, or characteristic that may provide scientific evidence of past or present life. Now, in astrobiology, biosignatures are extremely important because when we study distant planets, moons, we usually cannot go there. We often rely on light, atmospheric signals, surface chemistry, or patterns detected from far away. So the key question becomes, is this signal likely produced by life, or can it be explained by non-living processes such as chemistry, geology, or physics. So notice that I use the word likely. That is because science often works with probabilities. So especially when we are studying worlds that are light years away. So unless we can bring the samples back to Earth and study it in the lab directly, we must carefully weigh the evidence. Let me give you a few examples. Depending on the category, we have few categories here. We have for chemical biosignature, we have atmospheric gases. So on Earth, much of our atmospheric oxygen comes from photosynthetic organisms such as plants, algae, and cyanobacteria. For ozone, the ozone forms when oxygen in the atmosphere reacts with solar radiation. Since ozone depends on oxygen, it can also be an indirect clue. And next atmospheric, uh, next chemical biosignature is methane. So on Earth, methane is produced by microbes called methanogens, although methane can also come from geological sources. Our next chemical biosignature example is organic molecules. As I keep on mentioning, they are very important ingredients of life. So not all biosignatures are gases or molecules. Some are structures. So one example here is stromatolites, which are layered rock formations created over time by communities of microbes, especially cyanobacteria or photosynthetic bacteria. So these structures preserve evidence that life once interacted with its environment. So when we observe distant planets, we often cannot see individual organisms directly. Instead, we study light reflected, absorbed, or emitted by a planet. So different materials interact with light in different ways, which then creates unique patterns called spectra. Life can sometimes produce recognizable spectral patterns. So, for example, vegetation on Earth reflects light in a distinct way, especially in near infrared region, and this is called the vegetation red edge, which is a possible sign of plant-like life. And then also ocean surfaces can create patterns linked to water and surface conditions. Also, some pigmented organisms, such as, for example, halophiles, or salt-loving microbes, can also produce unusual colors and light signatures. So, in simple terms, a spectral biosignature is a pattern in light that may hint at the presence of life on a distant world. So that brings us to the end of our first lecture. Um, I hope this session has given you a strong foundation for the exciting talks that will follow after the break. But before we end, as promised, I told you that we are going to talk about life as we don't know it. Here's that 1% I mentioned. So here on Earth, life evolved under our sun. So for billions of years, every organism was shaped by Earth's chemistry, gravity, and the sunlight it receives. So even processes like photosynthesis were designed to utilize the wavelengths of the sunlight to sustain life. So this is life as we know it, which life that is written by the conditions of Earth. But the universe contains countless other worlds. So, what if life began on a planet orbiting a dim red star? What if it evolved beneath an ice covered ocean? What if it grew under skies with different colors, different chemistry, different seasons, or no sunlight at all? So, would it look like life that we know here on Earth? Would we recognize that kind of life? Would it breathe? Would it move? Would it grow? Or would it communicate in ways that we cannot yet imagine? So these are unanswered questions. These are unanswered questions. That is my way of making you appreciate what we study in astrobiology. And by the way, the image on the screen is not Earth, and this is not our sun. It is an artist's vision of the exoplanet K218b, which is a distant world orbiting a cooler, dimmer star. So can you imagine what kind of biology is brewing in this distant planet? So this is life as we don't know it. Somewhere among the billions of planets in our galaxy, nature may have written biology in another language. So personally, I believe that that is beauty of astrobiology. So it is not only the search for life elsewhere, but it is the search for life. This is the search for what life itself can become. And with that, I thank all of you for your attention. These are my references. And if I still have a minute or two, let me just share some recommended materials. As The Selfish Gene by Richard Dawkins is a good foundational book to learn more about the genetic world hypothesis. And also, I'm sharing this lecture that I was fortunate to attend 2 years ago in Denmark, which is entitled Are We a Unique Species on a Unique Planet? It is a very highly recommended watch if nabitin kayo dun sa sinabi kong lecture and you want to take a deep dive into astrobiology more. If you browse that page, you will see lots of different topics from big names in astrobiology. The talks were uploaded in this site. If you scan this, you can binge watch the videos if you want. They talk about exoplanetary research, synthetic biology, prebiotic chemistry, and all that. And with that, I thank you for your attention.
Okay, thank you so much, Miss Angie, for your wonderful presentation. If our audience can remember, schnapps, the pneumatic given by Miss Angie Macasinag, CHNOPS, carbon, hydrogen, nitrogen, oxygen, phosphorus, and silicon. It's really fascinating to hear about the different ways that life could have started, organisms that really like to live on the edge, and how biosignatures become really relevant in the investigation for the origins of life. I think that presentation really thoroughly prepares us for the foundation of the upcoming lectures here. So, if you have any questions for Miss Angie, please use the Q&A box above to have them answered later in our Q&A session. Before we move on to our next speaker, we'll be having a short 10-minute health break. Again, that's a 10-minute health break. Let's continue later at 10:10 a.m.
Maraming pangarap ang umusbong mula sa mga simpleng tanong. Mga tanong na humantong sa pagtuklas, inspirasyon, at paglalakbay. Matagal na nating tinutuklas ang kalawakan. Marami na rin tayong natuklasan dahil sa pagmamasid mula sa kalawakan. Ano na ba ang lagay ng panahon? Saan at kailan kaya tatama ang bagyo? Ano na ba ang kalidad ng hangin na ating nilalanghap? Pero mas malawak pa ang gabay na pwedeng manggaling sa kalawakan. Dahilan kaya tayo naglalagay ng infrastruktura at patuloy na kumukuha ng impormasyon sa ating planeta. Gabay tungo sa pangarap na makabangon at magkaroon ng mas maunlad na hinaharap katuwang ang agham at teknolohiyang pangkalawakan. Pangarap na abot-tanaw na sa pangunguna ng Philippine Space Agency o PhilSA. Itinatag noong 2019, ang PhilSA ang magtataguyod ng mga programa para tiyakin ang payapang paggamit ng kalawakan upang pangalagaan ang pambansang seguridad at suportahan ang ating pag-unlad. Bantayan ang kalikasan at tiyakin na makakaiwas tayo sa peligro ng mga sakuna at nagbabagong klima. Paigtingin ang pagsasaliksik at paggawa ng mga bagong teknolohiya. Suportahan ang pagsibol ng mga bagong industriya at pagsigla ng ekonomiya. Palawakin pa ang edukasyon at kaalaman ng publiko tungkol sa kalawakan at makipagkaisa at makipagtulungan sa iba pang mga bansa. Tinatahak na ng Pilipinas ang landas tungo sa makabago, mas ligtas at mas masaganang buhay para sa mga Pilipino gamit ang agham at teknolohiyang pangkalawakan. Isang karangalan ang pagkakataong maging bahagi ng paglalakbay na ito. Isang karangalan na isulong at panatilihin ang matatag na pangkalawakang komunidad na magpapahalaga sa mga mithiin ng mga Pilipino sa buong mundo. Isang bansang masagana, nakakaangat at may kakayanan, katuwang ang payapang paggamit ng kalawakan.
When the Philippines began building satellites, we started small. As small as a suitcase. Yet the stakes were high and the challenges were great. After the success with 6 of these small satellites, the Philippines is ready for something bigger. In December 2020, the Philippines kicked off the Multispectral Unit for Land Assessment, or MULA satellite project. Weighing around 130 kilograms, MULA will carry a true color camera that can capture higher resolution images, covering around 73,000 square kilometers of our country in one day. Data from MULA satellite will be used for addressing food security, disaster resilience, environment conservation, and protecting our national sovereignty. The Department of Science and Technology jump-started the funding of MULA. The project was jointly implemented by the DOST, Advanced Science and Technology Institute, and the University of the Philippines, Diliman, in coordination with the Philippine Space Agency, or PhilSA. 16 Filipino engineers who underwent a know-how training and transfer program in the UK are involved in building the satellite and are now part of PhilSA. The training capacitated our homegrown engineers, not just with knowledge, but the license to build more local commercial satellites in the future, leading the way for the creation of a robust and thriving space industry to support the country's space program. PhilSA will oversee the completion and launch of MULA and manage its operations. MULA in Ilocano means plant, pointing to the satellite's mission on food security. In Tagalog, it means point of origin or beginning, which symbolically signals the Philippines' foray into a new era in satellite development toward the vision of a nation bridged, uplifted, and empowered through the peaceful uses of outer space.
The Philippines is an archipelago of more than 7,000 islands, with rugged mountains and coastal plains, which make infrastructure development, particularly telecommunications, challenging. As an island nation, it is no stranger to connectivity issues. This is especially evident in far-flung areas that have yet to fully benefit from digitalization, whether in governance, services, or e-commerce. Although the country's internet and digital competitiveness have improved tremendously, there is still opportunity for growth and expansion so that more Filipinos can connect to the internet.
Kung pag-uusapan po ay ang internet connectivity po, ay hindi pa po ganun ka-stable ang connection po natin dito. In the other geographically isolated and disadvantaged area like the Kapanikian, ay hindi pa po sila nararating ng connectivity ng ating internet, maging ng anumang uri ng services when it comes to telecommunication.
Kailangan po namin tumawid ng dagat po, mga 1 hour po ang tatakbuhin bago ka pa makarating sa bayan para makapag-chat, makapag-internet.
Umuwi pa po kami Dingalan para po mabasa yung mga namin sa group chat, yung mga patawag po. Pag may patawag po si Mayor, pag may seminar, pag may meeting po kami sa DSWD.
Dahil isa kami sa pinakamalayong barangay dito sa Di Kapanikian, pagpunta pa lang namin sa bayan, gastos na.
Here's the good news. The Philippines is ready to explore satellite broadband technology with the help of inclusive access to satellite. The Philippine Space Agency, or PhilSA, the agency responsible for addressing all national issues and activities related to space science and technology applications, or SSTA, is guided by its powers and functions provided in the Philippine Space Act to promote and facilitate commercial space capabilities. One important downstream application of SSTAs is internet connectivity. With this in mind, PhilSA aims to bring relevant space technologies and associated policies to the forefront. To leverage the potential of emerging satellite broadband technology and address the connectivity gap, PhilSA collaborated with the Department of Science and Technology, Advanced Science and Technology Institute, and the United States Agency for International Development to implement the project Incentivize. Incentivize stands for Introducing Non-Geostationary Satellite Constellations Test Deployments to Improve Internet Service. Through this initiative, PhilSA hopes to put a spotlight on the space segment and support the country's inclusive and resilient economic growth.
PhilSA worked with USAID Beacon and DOST ASTI to engage communities and to collect data so we can assess the impact of the internet access and also address sustainability. Satellites can connect all of our islands. These technologies can bridge, uplift, and empower communities. We realize, therefore, that space is not that distant after all. All of us benefit from space capabilities in our everyday lives.
Because here in the Philippines, only half of Filipinos are participating online and using the internet. And so what we've been doing with PhilSA has really been testing and supporting the trialing of new internet connectivity technologies, particularly
Okay, thank you everyone. I hope everyone had the opportunity to refresh themselves before we dive into our next presentation. For our next topic, I would like to call Ms. Macasinag back to introduce our next speaker.
Thank you, Robert, again. It is my pleasure to introduce Dr. Paola Manini, which was my former mentor in Italy. So she is an associate professor at the Department of Chemical Sciences of University of Naples, Federico II in Italy. She obtained her PhD in chemical sciences from the University of Naples, from the same university in Naples in 2022. And in 2006, she became a researcher at the department's chemical sciences of the same university and has since been involved in numerous national and European research projects, authoring more than 100 scientific publications. So her research focus is on the chemical interactions of PAH that I have mentioned, polycyclic aromatic hydrocarbons, under extreme conditions, including space, to unveil potential implications in processes associated with the emergence of life. So her expertise ranges from organic synthesis to the analysis of complex mixtures, by applying different spectroscopic techniques such as NMR or nuclear magnetic resonance, EPR or electroparamagnetic resonance, attenuated total reflectance Fourier transform infrared spectroscopy, and Raman spectroscopy. Today, she spearheads a dedicated lab to understand the role of pigments, which is named after her, PM Chemistry, which I was a proud product of. She heads that research group, which was established 2 decades ago. And with that, let's all welcome Dr. Paola Manini.
Thank you so much. Thank you so much, Angie, for the kind introduction. I'm gonna share my presentation now. Okay. So first of all, I would like to thank the Philippine Space Agency for organizing this event and for inviting me. As Angie said, my name is Paola Manini and I'm Associate Professor at the Department of Chemical Sciences of the University of Naples Federico II. And today I will show you the recent advances in the search for the presence of life on planet Mars, also briefly discussing the scientific approach. For centuries, when the ancient people looked at the night sky, Mars was just a wandering red dot, a symbol often associated with war. But as our telescopes grew sharper and our probes began to bridge the celestial gap, that dot transformed and became the world, a world that looks quite similar to our own. So today, scientists are asking if we should go to Mars, and above all, was there ever someone or something already there? The search for life on Mars is not just a scientific mission. It is the greatest detective story in human history that can open to a new concept of life and mankind. The search for life on Mars is just about understanding the law of biology in the universe. To understand why we look for life on Mars, we must look backward 4 billion years. Early Earth and early Mars were siblings, almost twins. While Earth was developing its first oceans, Mars was not the desert we see today. Evidence from our orbiters and rovers such as Perseverance, Curiosity, Opportunity, and Spirit has confirmed that Mars once had a thick atmosphere. It had a magnetic field that shielded it from solar winds, and most importantly, it had liquid water. The presence of water is witnessed by numerous evident traces of ancient riverbeds, alluvial fans, and vast sedimentary basins that are present on the surface of Mars. To give you an idea, I've reported in this slide a collection of pictures taken from Mars, where is evident the past presence of water on the planet. The landing sites of the rovers Curiosity and Perseverance were the Gale crater and the Jezero crater, respectively, selected as sites in which ancient lakes persisted for millions of years. Here you can see in the inside the detail of the trace of the delta of the river flowing into the ancient lake occupying the Jezero crater, and also a simulation of how may have appeared the soil of Mars a lot of time ago. Traces of water are also evident from the mineralogic analysis of the Martian soil. As you can see here, the analysis carried out on the Jezero crater revealed the presence of hydrated silica besides carbonates and iron magnesium phyllosilicates. And when moving to the Oxyaplanum region, a high abundance of hydrated minerals was also detected. Based on this evidence, what we can deduce is that for a long window of time, Mars was not just habitable, it was a paradise for microbial life. But water is not the only part of the puzzle. To set the stage for the emergence of life, we also need the right ingredients. This is the reason why most of the Mars space missions have been also designed with the aim of identifying organic molecules possibly associated with life. And in particular now, I want to focus the attention on two representative space missions, the Mars Science Laboratory and the Mars 2020. Part of NASA Mars Science Laboratory mission, Curiosity was the largest, most capable rover ever sent to Mars when it launched in 2011. Curiosity set out to answer the question, did Mars ever have the right environmental condition to support small life forms called microbes? And 5 years ago, the most advanced robotic explorer ever sent to the red planet, the rover Perseverance, landed safely on the surface of Mars, ready to begin seeking the sign of past life. Here I want to, okay. Here I want to introduce you the rover Curiosity, the largest and most capable rover ever sent to Mars when it launched. As you can see, the rover is constituted by a series of instruments, including cameras, a weather station, and different spectrometers. Particularly relevant for the detection of organic molecules proved to be the robotic arm equipped with a drill, allowing for the collection of powderized rock samples of the Martian soil and sample analysis at the Mars positioned into the body of the rover used to analyze and identify the organic present in the soil samples. Here you can see a beautiful picture of the 42 drill holes made by the Curiosity drill when collecting the samples. All these powders have been subjected to the analysis operated by the SAM. This is a very complex and sophisticated suite of instruments constituted by a gas chromatograph, GC, a tunable laser spectrometer, TLS, and a quadruple mass spectrometer, as shown in this section of the instrument. The QMS and GC can work in tandem, operating in the GCMS mode for the separation for what concerns the gas chromatography and definitive identification for what concerns the quadruple mass spectrometer of organic molecules. The TLS captures precise isotope ratios for carbon and oxygen and measures trace level of methane and its carbon isotope. The sample analysis at Mars is used to analyze the samples as they are, so powders collected by the drill, or by using the wet chemistry kits that is constituted by special cups filled with liquid solvents. These solvents dissolve the organic molecules before heating them, allowing Curiosity to detect fragile molecules. In this slide, I have reported the rover Perseverance. Perseverance is an updated version of the rover Curiosity that has been improved also to analyze rock samples as they are without any manipulation. As you can see, Perseverance is equipped with different instruments such as MOXIE that produce oxygen from Martian carbon dioxide, different cameras, and a weather station. And we have also, in this case, an arm that is equipped with an X-ray spectrometer, Pixel, and the facility Sherlock. We will focus on the Sherlock facility because it's the one that is devoted to the analysis of organics. Sherlock is the acronym of Scanning Habitable Environments with Raman and Luminescence for Organics and Chemical. And it is an instrument that, as you can imagine, can perform Raman spectroscopy and luminescent emission spectroscopy directly on solid sample. Yes, in this case, regardless from what happened with Curiosity, Sherlock doesn't need to crush the rock and for the analysis. It scans the surface and creates a map. It can show us exactly where the organic molecules are located relative to the mineral crystals. So it's just like a sort of detective looking for the presence of organic directly on the collected rock. What emerged from this slide is that both the Curiosity and the Perseverance rover represent two sophisticated laboratories able to analyze the chemical composition of the Martian soil. And now I briefly show you the most relevant results that have been obtained. Starting from the results obtained from the NASA Curiosity rover, that has detected the different kinds of organic molecules on the Martian soil, including aromatic and aliphatic chlorocompounds and sulfur-bearing species such as thiophene. But most interesting was the detection of decane, undecane, and dodecane that are shown here on the top of the slides at the tenths of picomole level in 3.5 billion year old mudstone in the Gale crater. These are the largest organic molecules found on Mars to date, potentially representing broken down remnants of fatty acids, a key component of terrestrial cell membranes. The provenance and distribution of these molecules are of high interest in the search of potential biosignatures on Mars. But for sure, an outstanding result has been recently achieved with the discovery dated at the beginning of March 2026 of a new set of organic compounds, including aromatics, polycyclic aromatic hydrocarbons, benzothiophene, and nitrogen-containing heterocyclic compounds that may be related to the modern bases found in DNA and RNA. Overall, these results not only indicate the successful release of molecules preserved in ancient macromolecular or free organic matter within Martian bedrock, despite 3.5 billion years of diagenesis and radiation exposure, but represent another step forward for the demonstration of the past habitability of Mars. The data collected by the rover Perseverance after the exploration of the mudstone and conglomerate outcrops of the Bright Angel Formation in the Neretva valleys of the Jezero crater pointed out the correlation between the detection of polycyclic aromatic hydrocarbons and, as per Raman evidence, and the presence of minerals rich in redox metals such as nickel and iron, typically involved in the redox metabolism of primordial microorganisms. This analysis led us to conclude that the Bright Angel Formation contains textures, chemical and mineral characteristics, and organic signatures that warrant consideration as potential biosignatures, that is, a feature that is consistent with biological processes and then, when encountered, challenges the researcher to attribute it either to inanimate or to biological processes, compelling them to gather more data before reaching a conclusion as to the presence or absence of life. So to conclude, we can say that we stand at a crossroad. Through the chemical analysis of the sample analysis at Mars and the spectroscopic investigation of Sherlock, we have probably shed the light on the shadows of a habitable past, and we have found the water, the minerals, and the organic building blocks. The search for life on Mars is more than a hunt for microbes. It is a search for our own context in the universe. If life happened twice in one solar system, then the universe is teeming with it. If it happened only here, then we are even more responsible for the flame of life we carry. Mars is no longer a red hot, it is a mirror, and what we find there will ultimately tell us who we are. Thank you.
Grazie mille, Professor Paola, for that riveting presentation, especially since you had to wake up so early for this presentation. It's really interesting to hear about the current investigations for possible life on Mars. If you have any questions about the presentation, please do ask through our Q&A box. I'm sure Professor Paola would be glad to answer them later. For our third speaker, I would like to ask Dr. Hinojosa to give us the pleasure of introducing them.
Thank you, Robert. Our third speaker is Ms. Savannah Zigic, who earned her master's degree in biology of extreme environments under the same curriculum as Ms. Angie. She's also a cum laude graduate from the University of Naples Federico II in Italy. As part of her graduate training, she completed an Arctic microbiology field course at the University Center in Svalbard and conducted thesis research on the ecological responses of Antarctic diatoms to simulated melting and salinity change. She also holds a bachelor's degree in environmental science with a minor in geology from the University of Vermont, magna cum laude, where she worked as a field and laboratory technician investigating the effects of climate change and anthropogenic stressors on freshwater ecosystems. Her current research interests focus on extremophile survival, strategies in cold environments, the roles of polar phytoplankton in trophic and biogeochemical processes, and the impacts of climate change on polar marine microbial communities. Ms. Savannah Zigic, the virtual floor is yours.
Okay, thank you so much for that intro. Can everybody hear me okay? Can I get some thumbs up? All right, amazing. Let me just share my screen. Give me a second. Okay. So thank you so much to the Filipino Space Agency for the invitation to this seminar. I'm so happy to be part of this seminar and to be able to talk to you all today about the connections between life in Earth's cryosphere and our search for life on the icy moons of our solar system. So see, these are some of those icy moons in our solar system, and you can see in this picture their scale here compared to each other and also compared to the Earth. And there are, of course, many more icy moons in our solar system, but these are the ones we've visited with spacecraft, so we have real images and real data. So in this presentation, I'm going to give you a mini tour of two of these icy moons, and we're going to talk about why they matter in our search for life. But we're also going to keep coming back to the Earth in the meantime, especially its cryosphere, because studying these cold, icy environments here helps us understand what might be possible out there. And we're actually going to start by talking about the Earth, because I want to define for you all what we mean by the cryosphere. So Earth's cryosphere includes any environment where frozen water exists. So that could, of course, be ice in its many iterations, like sea ice or glaciers or ice sheets. But it could also include environments where we have snow, and even permanently frozen ground, which we call permafrost. And despite the incredibly harsh conditions found in these environments, including, of course, low temperature, but also in some cases you have high radiation, high pressure, and high salinity, these places are dynamic ecosystems and they're full of life. And when we study the life in these ecosystems, and actually more generally all life on Earth, scientists have identified 4 basic ingredients for life, which Angie talked a little bit about in her presentation. And I'll just go over them quickly now. So we're going to keep these ingredients for life in mind as a sort of checklist, a mental checklist, as we journey out to assess the habitability of our icy moons. So of course, our first ingredient is liquid water. It's the universal solvent that allows chemistry to happen. Then we have the essential elements. Remember those schnapps, the famous schnapps that we've mentioned before? These are the building blocks of our biomolecules. And then we, of course, need an energy source for our life. That could come from the sun in the case of photosynthetic microbes, but also, as Angie mentioned, we could have chemosynthetic microbes that use chemical reactions for energy, and we'll see many examples of those later on. And finally, you need stability over time. So essentially, you need these conditions to persist for long enough for life to emerge and evolve. So let's keep these things in mind, and we'll say goodbye to Earth for now, and we're going to travel out 5.2 astronomical units to Jupiter, which is our first stop. So although Jupiter is certainly amazing and fascinating in its own right, today we're instead going to focus on this little guy right here, one of Jupiter's moons, known as Europa. Now, compared to our own moon, Europa is just a bit smaller. It's about 90% of the size. And thanks to data from the Keck Observatory in Hawaii and the NASA Voyager probes, which gave us some of the first images and significant data for Europa, scientists figured out that the surface of the moon is made entirely of a water ice shell that's around 15 to 25 kilometers thick. And that shell is relatively smooth and free of impact craters, but it's also streaked by these long cracks all over. And that tells us that the surface is relatively young and dynamic and that there might be some kind of potential geology going on, which is very interesting. But what about beneath that shell? Well, that's where the NASA Galileo spacecraft comes in, which did a series of flybys of Europa between 1995 and 2003 while it was exploring the rest of the Jovian system, so the rest of Jupiter's system. And one of the most significant discoveries came from the probe's magnometer instrument, which can give us data about magnetic fields. And magnetic fields are one of the best tools we have for understanding what's inside a planet or moon. For example, on Earth, the motion of our molten liquid iron out of core creates a global stable magnetosphere that protects us from cosmic radiation. On the other hand, bodies without a molten convecting core like Mars, for example, or like our own moon, they don't have those global magnetic fields. Now, the Galileo probe detected a magnetic field around Europa, but it was different from the Earth's. Europa's magnetic field is super weak and it shifts around, it's dynamic. And scientists figured out that that's because the magnetic field around Europa is actually an induced magnetic field, which means it gets triggered by a different magnetic field, in this case, the massive one of Jupiter as it passes through Europa. And the only way to get an induced magnetic field is if you have some kind of conductive material inside the moon. And scientists calculated that that conductive material is most likely water-soluble ions. So that's a big hint that beneath that icy shell of Europa is a global salty subsurface ocean, which contains 2 to 3 times more water than Earth, and is probably around 4 billion years old. So because of that, Europa, along with a number of the other icy moons, are considered what we call ocean worlds. Now, by studying Europa's orbit, scientists also noticed that it's slightly elliptical, so it's not perfectly circular. So at times, as Europa orbits Jupiter, sometimes it's closer to the moon, or closer to the planet, sometimes it's farther away. And all the while, Jupiter's massive gravity is acting on Europa. So as it's getting closer and farther as it's orbiting, it's getting stretched and squeezed constantly by Jupiter's gravity. And that stretching and squeezing causes heating inside Europa's rocky core. And many scientists think that that internal heating could drive hydrothermal activity on the seafloor. And this discovery about potential hydrothermal vents is key because, like you saw in Angie's talk and here, we know that by studying hydrothermal vents on Earth, they're not only a potential place where life might have originated on Earth, but today they also host these remarkable ecosystems where you can find everything from microbes to complex animals. Now, before their discovery, scientists thought that the marine food web relied solely on photosynthetic organisms at the base, so phytoplankton and algae that use solar energy. But then they discovered these hydrothermal vents at the bottom of the ocean where there is absolutely no light, it's dark. So it turns out that instead of using sunlight as an energy source, these microbes use chemical energy from hydrogen, methane, and other reduced compounds released by these vents. And these chemosynthetic microbes form the base of the food web down there. So scientists, you can see, are very excited about the idea of hydrothermal vents on icy moons because that means there could be chemical energy and essential elements coming out of those vents that potential life may be able to use. So by studying these hydrothermal vents on Earth, we know that if all our hypotheses about Europa are true, inside the moon's subsurface ocean you could have liquid water, an energy source, essential elements, and stability, which if you remember our ingredients for life checklist, well, it checks all the boxes for life as we know it on Earth. But now comes the question of what exactly could that life look like, and how would we go about even looking for it or finding it? Well, at this point in time, we can't exactly drill through 20 kilometers of ice on Europa, at least not yet. So to understand what that potential life could look like, we turn to the closest environments that we can reach, which are those terrestrial analog environments. Now, these are not perfect copies, but they operate under similar conditions, like on the icy moons and other planets. And logically, many of the terrestrial analogs for the icy moons are found in Earth's cryosphere. For example, Blood Falls in Antarctica is one of the best analogs we have for better understanding Europa's reddish surface streaks. Scientists think that those streaks are cracks in the ice shell where fluids containing salts, sulfur, iron, and maybe even organics could be oozing up from the ocean below. Well, a very similar kind of process is happening at Blood Falls in Antarctica, where you have a brine which is rich in iron, salts, and sulfur erupting from the base of a glacier, from Taylor Glacier, forming this super dramatic rusty red waterfall. And the brine coming out of that waterfall actually originates from a subsurface lake underneath the glacier. And that lake has been isolated for millions of years. And it turns out that that subglacial lake is full of microbes. And these microbes are true extremophiles, polyextremophiles. They live in a low temperature environment. They also deal with high pressure, high salinity, and they live in complete darkness. So again, instead of using photosynthesis, these microbes use chemosynthesis to power their metabolisms. They oxidize various compounds and produce iron as a result. And then when that iron reaches the surface, it gets oxidized. So that blood falls waterfall is literally a liquid rust waterfall. So this example, blood falls, shows us that isolated icy environments can support active ecosystems using chemical energy alone, which is exactly the kind of thing we expect to be happening on Europa if we had that potential life. Now, on the other side of the world, on Ellesmere Island in the Canadian High Arctic, we find another analog for Europa, Borup Fjord Pass. And here you have a similar kind of thing happening. You have microbes beneath the ice that are oxidizing sulfur compounds and using that chemical energy. And they produce elemental sulfur as a byproduct, which you can see with that yellow stain on the surface of the ice. And what's super fascinating is that that sulfur produced by microbes has a different structure than sulfur formed abiotically, so formed through other physical chemical processes. So that means that that biotic sulfur could act as a potential biosignature that a future Europa mission might be able to detect. And in the same place, Ellesmere Island, this is where scientists discovered a special strain of Planococcus halocryophilus in the permafrost, so in that permanently frozen ground. And this bacterium is special because it can reproduce at negative 15 degrees Celsius, and it can maintain an active metabolism at negative 25 degrees Celsius. And that is a world record. It's the lowest temperature ever recorded for life doing either of those things. And that bacterium survives by using a suite of adaptations, including special flexible cell membranes, protective proteins, and it also has the ability to live in tiny pockets of salty brine within that permafrost. So this microbe helps us understand the true lower limits of life in terms of temperature, and it expands our sense of what potential life might look like on Europa or another icy moon. So as you've seen across all of these different terrestrial analog environments, Earth's cryosphere gives us a powerful window into what life might look like on Europa or the other icy moons, informing us about potential energy sources that life could use, about adaptations, the very limits of that life, and even potential biosignatures that life could give off that then we could detect with a future mission. And speaking of future missions, well, we have one on its way to Europa literally as we speak. This is Europa Clipper. It's a NASA orbital probe, which launched in 2024, and it will arrive at Jupiter and therefore Europa in 2030. So it will perform 49 flybys, close flybys of the moon, gathering data using a suite of remote sensing, but also in situ instruments. And Clipper will give us new detailed images of the moon in many different wavelengths, giving us brand new information. And it will also be able to sample and identify any particles that are ejected into space around the moon. And the main scientific goals of the mission are to, one, confirm the existence of the subsurface ocean, and then to also understand the nature of that icy shell, so the exact thickness, the type of ice, the composition. It's also meant to characterize the geology and the geologic activity of Europa. So Europa Clipper isn't going to exactly go looking for biosignatures in life. It's instead more of a habitability mission. So it's essentially giving us more data for that ingredients of life checklist. So that's it for Europa. Now we're going to say goodbye, and we're going to move on to our next stop, out into the solar system, another 9.5 astronomical units to Saturn, which is our next destination. And there we find our next icy moon, which is Enceladus. Now, compared to Europa and also our own moon, Enceladus is super tiny. It's only around 500 kilometers across. But it turns out that this little guy had a huge surprise in store. Because in 2005, NASA's Cassini probe did a flyby of Enceladus and took these images. These are real images of plumes erupting from the tiger stripes. These are 4 major fractures at the south pole of Enceladus. And Cassini actually flew into these plumes and used as many instruments to see what they were made of. And Cassini detected mostly water ice particles, along with a number of gases which we know are used by chemosynthetic organisms here on Earth for energy. And also, Cassini notably found phosphate, which is a biologically available form of phosphorus, which is rare to find. And Cassini even found simple organic molecules, which is very exciting. So right here we have extremely convincing evidence that, like Europa, Enceladus is also an ocean world. And that ocean world is full of some of those essential elements for life. Now, some of that plume material is actually ejected into space, feeding Saturn's outermost E ring. And by studying the material of Saturn's E ring, scientists found these tiny silica grains, and they are very specific silica grains that we've also found on Earth, where very hot water interacts with rock around hydrothermal vents. So, in addition to very convincing evidence of a subsurface ocean, we also have very, very convincing evidence of hydrothermal vents within that subsurface ocean. So based on everything Cassini found, Enceladus arguably gives us stronger evidence for habitability than Europa, because we've actually been able to sample that subsurface ocean and found evidence of all our ingredients for life, making Enceladus one of the most compelling places in the solar system for astrobiologists in the search for life. So if it were up to me, we would already be sending a spacecraft on its way to Enceladus right now. But it looks like we'll have to be a little patient because all missions planned to Enceladus right now are still at the proposal stage. But one of the most exciting proposals is NASA's Enceladus Orbilander, which you can figure out from the name. It means something that will orbit and also land on Enceladus near the tiger stripes of the South Pole. And there, the Orbilander would directly sample materials within the crevasses on Enceladus's surface, and it would use its suite of life detection instruments to actually look for biosignatures. So as opposed to Europa Clipper, which is more of a habitability mission, the Orbilander would be actively searching for life. But again, we'll have to wait a few decades before we see what Orbilander discovers. But in the meantime, the science definitely does not stop. Because if you remember our hydrothermal vents that we talked about before, well, there are some very special vents in the Aurora vent field in the high Arctic Ocean, which are permanently covered by sea ice. So that makes them one of our best analogs for the kind of environments we expect on icy moons. And what's especially exciting is that scientists are now studying which gases and organic molecules become trapped in the sea ice above the Aurora vent fields, and they're figuring out if these gases are produced by biological activity. And they are comparing those signatures to Cassini's plume data, which scientists are still actively mining through. So in other words, we can search for potential biosignatures right here on Earth right now, while we are waiting to send a new spacecraft to Enceladus. So in addition to this really creative and exciting science that's still going on right now, engineers are also busy inventing new technologies to be used on those future missions. And this is one of my favorite examples. This right here is NASA's EELS robot, the Exobiology Extant Life Surveyor. And EELS is designed to completely autonomously navigate hazardous, narrow, icy terrain, like the tiger stripes on Enceladus, for example, which it was designed and mined for. And engineers, of course, can't test it on the icy moons, so they use Earth analogs instead. For example, the mobility and navigation systems of the EELS robot were tested on the rough, icy slopes in the San Bernardino Mountains in California, while its sensors were tested by lowering the robot into a crevasse within the Athabasca Glacier in Canada in order to map the interior of the ice for the first time. So these tests not only serve the purpose of perfecting instruments for future missions, but they also give us these really cool, rare glimpses into hard-to-reach environments on Earth. And this is especially apparent when you look at another example. This is IceFin. It's a semi-autonomous underwater robot funded by NASA with the ultimate goal of being used for missions on icy moons one day, specifically Europa for this one. And to test IceFin, scientists have deployed it beneath the Ross Ice Shelf, where they captured this footage, this never-before-seen footage of this ecosystem that scientists didn't even know existed. So this is another amazing example of the discoveries being made about our cryosphere while these instruments at the same time are being tested and developed for future missions. So that's unfortunately all we have time for. I could talk about each of these moons for hours and hours, but I hope you enjoyed our little tour of Europa and Enceladus, and I hope you're also as excited as I am about the missions coming in the next few decades, because I really think they could genuinely change our understanding of life in the universe. So if you found this presentation interesting, I'd also encourage you to keep researching about our solar system's other icy moons, because it turns out that every single moon in this picture, scientists have evidence that they are also ocean worlds, and each one is unique in its own way. But I also Hope you see how essential Earth's cryosphere is to this work, since understanding how life survives in these extreme environments helps us think about what potential life might look like on the icy moons, while also, of course, providing testing ground for perfecting equipment to be used in future missions. And lastly, I just want to emphasize that unfortunately, these environments are changing rapidly. Glaciers around the world are retreating, permafrost is melting at unprecedented rates, Arctic sea ice is thinning and shrinking, and parts of the Antarctic ice sheet are destabilizing. So if we lose the cryosphere, we don't just lose these beautiful ecosystems, many of which are ancestral in current lands of many indigenous people, but we also lose a vital scientific resource, one that helps us understand how our own planet and prepares us to explore the icy moons. So as we continue searching for life in the universe, I hope we also continue researching and caring for the frozen places on our homeworld, because protecting Earth's cryosphere and exploring the icy moons are essentially part of the same scientific purpose of exploring the origins, evolution, distribution of future of life in our universe, which is, of course, astrobiology. Thank you, everybody.
Thank you very much, Ms. Zigic, for that very chilling presentation. Really interesting to hear how cold or freezing environments can possibly hold life. Again, if you have any questions, feel free to ask them through the Q&A box, as I'm sure our lovely speakers would love to entertain them. Last but not the least, for our final presentation in this webinar, if we could ask Ms. Angie to introduce them for us.
Thank you, Robert, and also especially thank you to my dear friend Savannah for preparing for this event. And for the next topic, it is also an equally we are also equally fortunate to have a fellow Filipino astrobiologist representing us from the United States. His name is Bernard Isayalo. He is a PhD candidate pursuing a PhD in astronomy at the University of Florida. He earned his Bachelor of Science degree in chemistry from the University of the Philippines, Diliman, after which he co-founded and worked at the DOST at the Radiation Research Center of the DOST Philippine Nuclear Research Institute. There, he was the lead on studying radiation protective chemicals. He was also a research associate at the Blue Marble Space Institute of Science, where he studied proteins involved in extremophilic properties of ancient microbes. Moreover, he has helped organize events to support the increasing local and regional interest in space and in astronomy through his involvement with the Space Generation Advisory Council. So his research at the University of Florida is on advancing a spectropolarimeter to detect a robust biosignature associated with the chiral properties of biological macromolecules and pigments. Currently at the breadboard phase, this instrument is being developed for use in life detection surveys of astrobiological targets. Let us all welcome Sir Bernard.
Hello. Thank you very much, Angie, for that very kind introduction. First things first, I just want to double check that I am audible. Yes. Perfect. Okay. Let me just share my screen then. This one. Okay.
Mr. Bernard Isilo, I would like to inform you that you are currently not audible, or your video presentation is also not active or moving. Mr. Bernard? Okay, apologies to our audience members here. We are currently trying to fix this technical difficulty with Mr. Bernard. While we're waiting for these technical difficulties to be fixed, we are yet again notifying our audience members to post any questions in our Q&A box. Should it be within our topics that are discussed, you may specify for each speaker if you would like to specify which question you would like to ask, or if you would like to throw a general question for our panel of speakers. Let's do a couple of shout outs in our audience members. Shout out to Mr. Errol John O. Ayon from Nuestra Senora de Guia Academy. To Lindsay Angeline Abelar from the Manuel S. Enverga University, Lucena. Mr. or Miss Kaino Polinar from the University of Southeastern Philippines. We have here David Jonathan Interino, Polytechnic University of the Philippines. Joanna Paula ex-Conde from the University of Southern Mindanao. We're happy to accommodate all of you from all over the Philippines. Kyla Abilar, Rizal Technological University. Kevin Ramos from the University of the Philippines. Ian Gamad from the Department of Agriculture. Jean Jimenez from Centro Escolar University. So again, please hold. We appreciate your patience during this time as we are trying to detect this difficulty and fix it. There was a question about sharing the file for our lectures. We would like to say that we apologize if we're not able to provide copies of the lecture slides for our research speakers. If you have any specific questions or would like to further clarify on the topics discussed, please feel free to send them to the Q&A box and we'll raise them during the Q&A session. Thank you for understanding. However, to accommodate the request for the lectures being reviewed again, we would like to notify everyone that the recording would be available for viewing but not for downloading. In order to accommodate some time during our technical difficulties, let's have a 5-minute bathroom break for everyone. We'll see you at 11 o'clock.
Hello? Can you hear me? Okay. Sorry, I didn't know what happened there. I'll probably just close my camera to be safe.
Hello, Mr. Bernard. We're currently in a bathroom break to accommodate the technical difficulties. Feel free to
Okay.
fix anything on your end until 11 a.m. when we would start the presentation once again.
Yeah, I apologize. I didn't know that I was disconnected. I was
That's
talking.
perfectly fine, Sir Bernard. Though we would like to ask you to, again, please reshare your screen as it is gone when you got disconnected.
Yeah, sure, no problem.
Thank you. Again, Sir Bernard, we'll be starting at 11 a.m. as we told the audience, the participants, to have a bathroom break until then.
Okay, sounds good. Can you see my screen?
Yes, the screen can be viewed. Thank you.
Okay, thanks.
Okay, I hope our participants were able to refresh themselves. I am asking once again, Sir Bernard, we may now start with your presentation.
Thank you so much. I apologize for that technical interruption. I had no idea. I just kept talking. So I appreciate you guys for reaching out to let me know that I was actually disconnected. Anyways, so maybe I can begin again. I'd like to thank everyone for attending these series of talks. All the talks have been very interesting for me so far. I'd like to thank the Philippine Space Agency for inviting me, particularly to Angie. So without further ado, my topic. Okay, there's my laser pointer. Okay, I would like to begin my talk by showing this picture here. This picture here on the right is that of Earth, recently taken during the Artemis 2 mission, as many of you may be familiar with. Artemis 2 just concluded about 2 weeks ago. I think this is a good reminder that we have known only one planet that has life. Whether that means present or past life, we have really only known um, our own Earth. And for thousands of years, humanity has always been interested in finding life on other worlds. And in this talk, we will explore some of those approaches we have conducted to detect these signs of life. And you'll see that we've been scientifically looking for life for many decades. We are now at a very promising point in time where we have all these brand new telescopes, rovers, laboratory experiments, and missions that I personally will not be surprised if we would find life very soon. Definitely, I think within our lifetimes. But first things first, now just so we are on the same page here, this is basically a review from a few lectures ago. Angie mentioned as well, and that is biology is the study of origin, evolution, and the distribution of life in the past, present, and the future of the universe. And this topic, methods to detect signs of life, really is related to the distribution of life in this universe. And as you can see, this definition is pretty broad. It speaks to how multidisciplinary this field is, to the point that somebody like me, who graduated with a bachelor's in chemistry, from the Philippines can actually, not really move fields, but in a sense, conduct research in a graduate program of astronomy, only because of astrobiology being so interconnected. All our different fields have some sort of contribution to this wonderful field of astrobiology. But the story of astrobiology really began in 1976, exactly 50 years ago. Just to set the stage a little bit here, 1976, the two superpowers of the world at the time, the Soviet Union and the United States, were at what we call the Cold War and the space race. And with that, with sending people to the moon, sending a bunch of satellites into space, astronomy and space were very much in the minds of the public, at that time. And with it came the interest in the question of, are we alone? Is humanity alone in this vast cosmos? People were also thinking about the next target in terms of, space exploration. And for many people, this next target was Mars. And in fact, in 1976, NASA sent a couple of landers to the red planet, shown here. This is a replica. This is not on Mars. This is not a Martian, but this is a replica of one of the Viking landers. And this is a scientist by the name of Carl Sagan. A lot of people here might be familiar with that name. But the Viking 1 and Viking 2 missions were really the first ones to reach the Martian surface and the first ones to search for evidence of life on Mars. This is, I believe, the first photo that the Viking landers took while on Mars. So the Viking landers landed on Mars. It did its experiments, and the results the results from the Viking missions are actually still a matter of debate to this very day. This is one of the early papers that came out of the mission, and I like to just highlight this phrase, which basically summarizes the position that many scientists have with the Viking results, and that the results can be explained as non-biological, meaning through geology, chemistry, or physics instead of pure biology. Now, another part of this paper that I thought was interesting and a little funny that I want to highlight is this. So there were no little green men, in quotes, and a fair segment of the public very likely anticipated such findings. This was included in a scientific paper in 1976, and I think that just gets you in the headspace of the public at that time. Nobody has ever sent out a space mission to any other planet or any other world to detect signs of life. So nobody really know what to expect. Maybe a lot of people expected when they landed on Mars, suddenly there's a lot of, aliens walking around, and we'll get a picture of those aliens. Sadly, Viking proved that that was not the case. So the hype around astrobiology died a little bit after it became increasingly clear that the results were probably non-biological, until about 20 years later, where a meteorite called ALH 84001 suddenly made its way into the news. And for multiple reasons, scientists claimed that there are microfossils and other evidences of life in this meteorite. This became so publicly known that the US president at the time, Bill Clinton, actually had to make a statement in front of the White House saying that this can surely be one of the most stunning insights into the universe that science has ever uncovered. However, from the beginning, there was already reasonable skepticism about the results here. Other researchers refuted the conclusions of the paper. There wasn't really any reproducibility experiments, for example, with their claims. And up to this day, the handling of that paper in 1996 caused a lot of debate, on the ethics of science communication. These sorts of results have to be conveyed to the public. On one hand, it was unfortunate that the findings were publicized and sensationalized, but on the other, it did increase public awareness and sparked interest, leading to NASA to prioritize research in biology. In fact, in 1998, that's just 2 years later, NASA established the NASA Astrobiology Institute. And looking back, it is really difficult to believe that missions that we have today, like Curiosity and Perseverance, none of them would probably happen if it were not for the McKay paper and the ALH 84001 meteorite. So now this is sort of the landscape of astrobiology. I show here some of the telescopes and missions that are in various stages of implementation. I note that a lot of these instruments have more than one function besides searching for life. ALMA, for example, the Atacama Large Millimeter Array and the James Webb Space Telescope are extensively used in astronomy research as well. But if you notice, they can actually be grouped into two distinct groups. One that I would call remote sensing missions and one that I would call in situ missions. And the differences between them are relatively simple. For in situ missions, they are measured basically in the extraterrestrial environment itself. We send out orbiters, we send out rovers, and we look for specific organic molecules, isotopes, minerals, gases, or physical evidences like cellular structures, stromatolites, I think Angie mentioned that as well, and growth, On the other hand, remote sensing deals with remote observations, in particular, spectroscopy. We use telescopes here on the ground or in space to look at other worlds. And this is actually, while we can do this for all astronomical objects, we can only, for exoplanetary research, we can only really use remote sensing missions because in situ missions to places outside our solar system is unfortunately technologically impossible for the foreseeable future. And because it uses spectroscopy, we look for biosignatures like gaseous molecules, like oxygen and methane, as also discussed in the previous lectures. So now, even though I am an astronomy PhD student and I love my telescopes, there's just something about actually going to a planet and doing chemistry experiments, which of course brings me back to my chemistry undergrad days. And a great example of these in situ measurements just came out literally last week with a Nature paper from Amy Williams and her collaborators. Dr. Paola Manini talked about this a little bit as well. Amy Williams is a professor at the University of Florida. I actually got to talk to her a little bit last Friday, and believe me, her inbox is crazy these days. But anyways, this paper reported the in situ detection of more than 20 organic molecules using the instruments on board the Curiosity rover on Mars. And what they did was really cool. They drilled a hole into a rock. They extracted the molecules using a high pH reagent and then heat to volatilize the molecules into a gas. And then they placed that molecules into a gas chromatograph, which basically separated out those molecules into its individual components. They analyzed those components, and these were the results that came out of that analysis. And if you notice that there are so many organic molecules, not only that, there are what we call aromatic organic molecules, which are particularly interesting in the context of life. So this paper gets me really excited because it does seem like there are a lot of complex organic molecules on Mars. Of course, the question now is, how did it get there? It can be exogenous, meaning from meteorites that carried the molecules there, or it can be indigenous, basically from Mars, from the geology in Mars, or maybe from life. We hope to answer this question in the succeeding missions. I believe ESA will be sending out the Rosalind Franklin rover in the next few years. So we just have to stay tuned with this story. Now, going beyond Mars and the solar system, perhaps even more exciting is that we can use telescopes like JWST to look at exoplanets. These are planets outside our solar system, way beyond Neptune, way beyond Pluto and the Kuiper belt, hundreds of light years away. There was a famous paper, maybe infamous paper, published by Manusudan and his collaborators last year that claims to have detected a specific molecule called DMS or dimethyl sulfide in the spectrum of the exoplanet called K218b. This is a famous exoplanet. Angie mentioned it in her talk as well. DMS is special because on Earth, this molecule is only produced, mostly produced by marine phytoplankton. And what they did is that they collected the light coming from a star when the planet was in front of the star. So the light passes through the atmosphere also of the planet, which means that they could theoretically measure any gas molecules that absorb that light. And the resulting absorption spectrum is shown here on the left. And through modeling, they were able to fit the spectrum, I should say, with a few molecules, including DMS and DMDS, which is dimethyl disulfide. This sounds really amazing. They seem to have detected DMS in an exoplanet 120 light years away, but not so fast because atmospheric characterization for a specific exoplanet hundreds of light years away is really hard to do. As you can see, while there might be peaks there, the error bars are pretty big. We're talking about really, really small wiggles, quote unquote wiggles, spectrum of a star. So it's really hard to actually make robust conclusions out of that. And some authors believe that the spectra can actually be fit by just a flat line, unfortunately. However, this is such a new field, this was not possible 5 years ago. And the fact that they are arguing about these tiny wiggles in the spectrum should give us hope for improved measurements in the future. So similar to the in situ measurements that we discussed before, we just have to stay tuned for future measurements and in hopes of having improved data. So there are many signs of life that we've talked about with various levels of credibility, as well as ease of measurement. Here, I just quickly introduced the concept of the ladder of life detection. This is a concept that ranks the biosignatures in order of increasing strength of evidence of life, but also decreasing ease of measurement. They go hand in hand. On the left here is habitability. This is the easiest, quote unquote, easiest to measure, and it's a good first step. Things like the habitable zone, temperature, chemical disequilibrium, this all goes into the concept of habitability. And on the far right, the most, convincing evidence of life is if we could theoretically observe Darwinian evolution. Unfortunately, this is not practical under mission constraints. It takes millions of years for evolution to occur. So while these are good for biologists because they could study fossils and stuff, they're not so good for astronomers because we only, basically collect light or send out rovers to these far planets. So we're left with others. These are proxies, which are further down the ladder in order of decreasing of strength. And they include features like metabolic byproducts, potential biomolecular components, structures with implied function, metabolism, growth, and reproduction, including some morphological evidences. Okay, so I presented many concepts as well as the big missions from NASA, but several institutions are also working on building the next generation of instruments that might be able to help detect light. And one of these missions is being built by our team here at the University of Florida. We have built a few prototypes of this instrument shown here on the right. This is the first version, the second version, and the third version. These laboratory measurements, this is the third prototype from the last slide. I worked on this instrument the most. This will be basically the instrument I used for my PhD dissertation. And the design of this instrument is relatively simple. It has no moving parts. They're all just basically lenses and different glass components laid out in a cage system like this. This is the camera that basically takes the images, and then this is the telescope that collects the light from a target, say, here on the right. And what this instrument tries to measure is circular polarization of light. Um, and that's because we're trying to probe for a specific biosignature called homochirality. And what homochirality is basically is the fact that terrestrial life prefers only one mirror image, where amino acids are only, in biology at least, left-handed, sugars are right-handed, alpha helices in proteins are right-handed, and so is DNA. This also goes in all levels of biology, from the simplest molecules to the largest supramolecular assemblies. So our instrument tries to probe these structures without actually having to know what the identity of those molecules are. This is an example of that spectrum, and it basically tells us the large scale structure of our photosynthetic pigments in plants. And while we are using this instrument mainly in the context of astrobiology, there are other more earthbound use cases for our project. We are also in the process of commissioning the next generation of this instrument, and we are excited for this one in particular because it's smaller and it can be actually attached to telescopes. So we're excited for that. Maybe this year this will come online, and we'll finally be able to look at far planets. Instrumentation is not a individual effort, so there's a whole team that works on this, and I just show some pictures of them over here. But with that, I appreciate you all for listening to my talk. Feel free to reach out if you have any other questions. I put my email as well as my LinkedIn profile. Thank you very much.
Thank you so much, Mr. Bernard, for that enlightening presentation. Fascinating to hear the history of how we have explored astrobiology, even from way back, and the current processes that we are using to find signs of life. Now that the presentations have ended, please join me in giving our esteemed speakers some love in the chat box. Please do express your gratitude to them. So I hope everyone was able to learn a lot from their discussions. As I'm sure everyone has a burning question, we will now be opening the floor for our Q&A session. If you have any questions, feel free to send it through the Q&A box found at the top of Microsoft Teams. You may ask the question specifically to one of our speakers in the webinar.
So for now, we'll be starting with some questions sent through our discussion forms. I think this one is for Miss Angie. This Danielle Kalikaran from West Visayas State University asks, from what I have known, astrobiology is not a known career in the country. What do you advise to Filipino students who want to pursue a career in this field, but also cannot afford to study or work abroad? Where should we start, and how can we grow as future Filipino astrobiologists? Yeah.
Yeah, thank you for that question. So I think I'm going to share my own personal experience, and I would also like to share the stage for a fellow Pinoy astrobiologist here. Also, if he can share his own experience in his journey towards because as I've mentioned, right now he is currently a PhD candidate. By the way, congratulations, Sir Bernard, for recently passing his PhD candidature. Anyway, when it comes to I admit that dito sa Pinas, mahirap talaga. It's really very hard to pursue basic science, especially that we have limited resources. We have to admit that. So, as I've mentioned, astrobiology is not just reserved for biologists. In fact, if you are currently pursuing, say for example, geology, you also have a place in astrobiology. So in a way, you can connect what your current field is and somehow find your path in astrobiology. So personally, I started as a biologist. So my professional background is my academic background is in biology, but I really love astronomy. So I'm doing astronomy on the side. I'm watching YouTube videos, as most of you probably do. I'm watching astrophysics video and all that. So while I'm pursuing biology, I do not leave behind my first love for astronomy. And then somehow I found an opportunity to apply for graduate study in Italy, and they offer astrobiology. And then, of course, I am not actually financially capable to pursue that kind of study, especially that I need to pursue that abroad. Especially in Europe, it's very expensive to live there. But somehow, fortunately or luckily enough, I was able to get accepted to the scholarship program of the Philippine Space Agency called AD-ASTA. So if you have if you are currently in a different field right now, don't feel sad because you can somehow manage to, if you really want this field, astrobiology or any other field, tiwala lang, you will find your path there and work hard.
All right, thank you for that, Ms. Angie. If Mr. Bernard would like to share his experience as well.
Yeah, definitely. I definitely echo Angie's statements there. It's really difficult to pursue basic research and especially astrobiology because it is such a niche field. But as she said, it's so multidisciplinary that no matter what your field is, where you start, your knowledge can actually be helpful and useful for astrobiology. So in my situation, I graduated from BS Chem in UP Diliman in 2018. But you know, just even before graduation, I think I already knew that I wanted to do space research and astronomy. So what I did was I actually, took it upon myself to cold email a lot of professors, professors from all over the world, in case they had any research positions for me. You miss all the shots you don't take. Um, so luckily there was one who basically responded, and we went on a lot of Zoom meetings. It was in 2021. This was in the middle of COVID, but also she was American, so there was no way to meet her in person. Um, and she encouraged me to pursue a PhD, and she actually connected me with a bunch of other professors in Europe and in the US. And then 2 connections later, I finally connected with my current advisor here at the University of Florida. He encouraged me to apply at the astronomy grad program, and luckily I got in. And the great thing about PhD programs here in the States is that it is usually fully funded, both in terms of tuition, maybe not fees, but it's a small part compared to the tuition, but also a stipend that's not a lot, but it allows you to basically have the necessities that you need. So you don't need to be financially secure to be able to pursue a PhD, at least here in the States. Obviously now, maybe it's a little harder to get the funding with all the, let's say, issues that are going on. But there are still a lot of opportunities out there if you know where to look. So I encourage everybody to just continue being curious, read a lot of papers. If you find something that's interesting, all those researchers in the author list, they're all really excited to talk about the research with you. So email them. They have their emails at the bottom of the papers almost every time, and you never know where you'll end up if you just email them and ask them a bunch of questions. So that would be my advice.
Thank you so much, Mr. Bernard. So we now have a question for Professor Paola. Miss Angelina Kieran from the University of Santo Tomas. Could extremophilic microorganisms such as fungi play a role in future Mars colonization, perhaps in terraforming, radiation shielding, or life support systems?
Okay, thank you. Yes, it's a very interesting question, this, because uh, basically we think that some very simple microorganisms, such as fungi, has a very special power inside, because I mean, regardless the fact that they are very simple in the structure, in the biology structure of the cellular environment, we should consider that they have developed and survived in very harsh conditions. And so probably this is the secret of this kind of simple microorganisms. So basically, we can imagine the fungi and also bacteria has the possibility to be used as primer in the development of a new colonization of planet different from ours, because they possess very interesting properties, not only in terms of survival to very peculiar conditions, and I mean conditions in which we can have very low and or very high temperature, but also they can survive in condition where we can have a severe impact of radiation on a specific environment. And there are many evidence on the ability, especially of fungi, to survive under these conditions, because they can develop a sequence of safety weapon that they possess to fight against these extreme conditions. And so the hypothesis that this kind of microorganism can be very useful in this sense is very timely, and there are many scientists, many research groups that are working in this direction, trying to also establish, to investigate how this kind of microorganism can survive under extreme conditions.
Thank you so much, Professor. Hopefully that the question has been fulfilled. The next question is for Ms. Savannah. This is from a PhilSA personnel. From your experience working in cold environments like the Arctic, can you share the biggest challenges in collecting samples and keeping them intact for lot analysis?
Sure, thank you for the question. So I have a little bit of experience working in the Arctic, so in Svalbard. And of course, with any kind of field sampling, you need lots and lots of planning in advance. But when you're in the Arctic, or I suppose in any extreme environment, you need to plan that much more. For example, in Svalbard, they're known for their polar bears. So that was something we always had to keep in mind. Everybody receives polar bear training. Everyone carries a rifle and a flare gun, and you always, before sampling, scout to see if there's any polar bears. And you always have a scout while you're sampling looking for polar bears. So that's just an additional thing you have to keep in mind. And then in terms of sampling itself, one big thing is a lot of these environments are pristine environments and they've been isolated for millions of years. So you want to do everything you can to prevent contamination of those environments. For example, I was sampling in a cryopeg, so a lake underneath the permafrost that had been literally isolated for millions of years. So we had to sterilize all our equipment. We had to make sure we were being as least invasive as possible, only sampling as much as we needed to, stuff like that. And then another thing is it's really cold. So I had a problem like in the field, we had to take that sample and we had to filter it. So push water through a syringe into a filter, and it was so cold my hands stopped working. So that's something you have to also keep in mind. So working in an environment like that, it takes extra planning, but also like a positive attitude too, and like a certain amount of like gusto, which is not hard, you know. It's really fun doing these kinds of things. So yeah.
Thank you so much for that wonderful insight, Ms. Savannah. So this question is for Mr. Bernard. Mr. Neil General, I hope I pronounced that right, from the Philippine Science High School Cordillera Administrative Region Campus asks, is there a possibility of life utilizing different chemicals to thrive on, such as using silicon, ammonia, etc., instead of the usual water and carbon? If so, how likely? If not, why?
That's a great question. Um, if the question is if life can use these atoms, then the answer is always yes. Life will always find a way to adapt to its environment. Whether or not it's probable is a different story. With silicon, I think there might be two reasons why this might be improbable. One reason is astrophysical in nature, astronomy, and one is more biochemical. Um, in the astronomy perspective, all the elements in our universe, with the exception of hydrogen and helium, are produced in or by stars. Carbon, and sorry, as you go up the periodic table, or should I say down, as you go into higher atomic weights, it becomes harder and harder to produce. And silicon is further along the periodic table than carbon is. So in terms of abundances, there's a lot more carbon in the universe than silicon, right? As far as I know, there's about 10 times more carbon in the universe than silicon. And life would most likely use what's most abundant, and carbon would probably be the most likely atom for biochemistry. In terms of the biochemistry aspect, silicon, just because it's lower on the periodic table, it's a bigger atom. So it's a lot harder to produce biomolecules, which tend to be compact and have to have these various arrangements for them to function properly. Carbon, is small. It can do that fairly easily, but it might be a little bit more difficult for silicon to take the place of carbon. That being said, if there is just silicon and life has to use it, then I won't be surprised that it can find a way to do that. I'm just saying that in terms of astronomy, biochemistry as we know it, it's a little improbable to imagine.
Thank you so much, Mr. Bernard. Hopefully that answers our participants' questions. So we have questions from the live chat. So this one is from Charlie Tavis. Thank you po, Ms. Angeline, for the insightful presentation. So here's my question po. How can extreme environments in the Philippines be leveraged as analog sites for astrobiology? And what are the implications of astrobiology research for countries like the Philippines in terms of scientific development and environmental research?
Thank you for that question. So as I've mentioned, we have a lot of diverse environments in the Philippines, and some of those diverse environments actually are extreme environments. So here in PhilSA, we want also to try to utilize those kinds of environment and use them for astrobiology research, hopefully, and also some applied sciences like biotechnology and such like that. So given that we have a lot of potential here in the Philippines that we can actually use as analogs for studying life, possibility of life out there. So in terms of implications for Philippine research, actually, if we ever found a way to establish this kind of biology of extreme environments, as you say, if we were able to establish that kind of research, we can contribute meaningfully to the basic research, not just actually in the country but globally. So we have now with the advent of space technologies, numerous successful programs targeted not just for space research but also in advancing future of human in space. So the implication of those kinds of research utilizing extreme environments for as analogs, we can actually contribute to that kind of global pursuit to hopefully establish human settlement in space. So yeah.
Thank you so much, Ms. Angie. So we have another from Mr. Steve. For Miss Savannah, does the icy moons have atmospheres similar to Earth? And would that be relevant in the development of life? Also, do we have evidence or basis to ground the expectation if the life there is simple or complex?
So, as far as we know, the icy moons do not have significant atmospheres, with one exception, which is Titan, one of Saturn's moons. But Titan has a different atmosphere than on Earth. It's got, I think, mainly nitrogen and a little bit of methane. So slightly different composition from what we have on Earth. And also, like Angie mentioned, because of that bit of methane in the atmosphere, instead of liquid water on the surface, you have liquid methane and ethane. So essentially, rivers and lakes and such, and rain and clouds made of methane and ethane. And yeah, so any kind of life on Titan's surface probably wouldn't be water-based, so that's kind of going outside of our what's known, what kind of, like on Earth, what we expect life to be like. So it's tough to answer that question. It would be life as we don't know it on Titan. Yeah.
Thank you so much, Miss Savannah. So do you have any more questions? I have a question for Professor Paola. So, since Mars gets a lot of radiation that can break down organic materials, how does that affect our chances of finding biosignatures? And does that mean it's more promising to look for signs of life below the surface instead?
Thank you for the question. Yes, because this is a very a great problem of paramount importance because as you mentioned, we are, for example, taking into account the fact that Mars is as not a protective shell against high energy radiation. And so when we look for biosignatures, the presence of organic molecules on the surface, this could be a very great problem because we know that this kind of radiation can completely destroy many organics. And there are two different approaches that are currently under investigation in this moment. From one side, the importance of the mineralogic environment in acting as a protecting factor against the destroyment of this kind of molecule. For example, there are evidence that some kind of sulfates can, in a certain manner, protect organics. And so if we consider that this kind of molecule are completely distributed, absorbed on the surface of this kind of mineral, we can imagine that they can be preserved against high energy radiation thanks to the role played by this kind of mineral. And another approach is exactly what will be the focus of the next ECA mission, the ExoMars that will be launched, we hope, on 2028. And regardless from the previous one, the ExoMars mission plan to collect sample by drilling 2 meters in depth. That is more in depth with respect to the previous samples that have been collected and analyzed by Curiosity previous mission. So we can imagine that by reaching very in depth this kind of sample, the radiation impact should be minimized. And so probably we can bypass this kind of problematic and have the opportunity to get and to analyze directly the organics as they are without any kind of effect by the high energy radiations.
Thank you so much, Professor. So we have another question for Mr. Bernard. Can you explain more about agnostic versus traditional biosignatures? Outside our solar system, what element is most likely to be the building blocks of life?
Okay. I'll try. So Traditional biosignatures, as I understand it, basically focuses on what we know about life on Earth, right? In terms of the biochemistry, in terms of the gases that they produce, oxygen, methane, the disequilibrium between the two molecules, that sort of thing. Agnostic biosignatures, on the other hand, tries to be agnostic, tries to not have any assumptions before going into any life detection mission. So our instrument, for example, is a type of agnostic. It tries to detect these agnostic biosignatures because we don't really assume that life, let's say, is carbon-based or whatever. We assume there's one assumption, and that is life tends to only use just one chiral form of a molecule, one mirror image of a molecule, for whatever reason, right? It could be just it's more energetically favorable for biology to use just one-handedness. For enzymes, for example, or whatever life outside of Earth is the equivalent of enzymes for life outside of Earth. They have these selectivity that only is possible with one-handedness of molecule. So yeah, agnostic biosignatures try and have as little assumptions as possible before measuring anything. And then the second question, can you repeat that? Sorry.
Yes. Outside our solar system, what element is most likely to be the building blocks of life at the moment?
Right. I think this is related to the question that we had before, and that because the physics of the universe we think is the same, that means that the abundances of the elements would be about the same, hydrogen, helium, and then you go up the periodic table and such. So, life on Earth uses carbon, and as far as we know, there's really no reason for life to use anything else. Of course, that doesn't preclude any possibility of, say, a specific region, say in a star-forming region that has an enriched amount of silicon, for example, or whatever. But in general, I still think that carbon-based life is most likely. Other people might disagree, and I think that's what's great about astrobiology is that we can all talk about it, and we can all discuss the science behind it, and we all generally come to some conclusion, or if not, then it's the next generation's problem, right?
All right. So thank you very much to our dear speakers and for the participants who asked their questions. And I would like to apologize, but in the interest of time, we can no longer accommodate further questions. So if you have any more questions, you can email your questions to ifnb@philsa.gov, and we will coordinate this with our resource speaker. That concludes our question and answer session. I join me to give our esteemed speakers a round of applause.
Okay, thank you so much to our speakers. We would like to take the opportunity to present the certificates of appreciation for our esteemed speakers. In the certificate, so this certificate of appreciation is presented to Ms. Angeline Macasinag, Dr. Paola Manini, Savannah Zigic, apologies, Bernard Izialo, for sharing their expertise and knowledge as a resource person during the HugNayan, the search for life beyond Earth. or apologies, during the HugNayan Online, the search for life beyond or Earth, given this 28th day of April 2026 at the Philippine Space Agency, 11 Eastwood Avenue, Bagong Bayan, Quezon City, Philippines. Signed by Dr. Reynable C. Reyes, Director 4 of the Space Science Missions Bureau, Dr. Mark Cesar R. Talampas, Deputy Director General of Philippine Space Agency, and Dr. Gay Jane P. Perez, ad interim Director General of the Philippine Space Agency. Please give our speakers a round of virtual applause. So along with this, we would also like to ask for a photo opportunity with our esteemed speakers. So if you are able, we are requesting you to open your cameras. We would also like to acknowledge the presence of Ad Interim Director General, Dr. J. Jane Perez, Deputy Director General for Space Science and Technology, Dr. Mark Cesar Talambas, and Director of Space Science Missions Bureau, Dr. Reynable Reyes. We are also inviting them, if they are able, to please open their cameras for a photo opportunity with our speakers. And we would also like to acknowledge the presence of Dr. Ernest Macalalad, the Chief Science Research Specialist of the Interdisciplinary Space Missions Development Division of the Philippine Space Agency. Okay, if you're ready, let's have our photo. 1, 2, 3, smile. Okay, another picture, please. 1, 2, 3. Okay, thank you so much to everyone who has tuned in with us today, to all the students, enthusiasts, and researchers who are part of different universities, organizations, and institutions. Wherever you are, we hope we have helped you quench your thirst for curiosity on the search for life beyond Earth. Again, we are asking everyone to please stick around as we will have a photo opportunity with our audience participants. However, first, to give our closing remarks, please allow me to call on astrophysicist and director of the Space Science Missions Bureau of the Philippine Space Agency. Please join me in welcoming Dr. Reynable Reyes.
Thank you, Kenneth. Thanks, everyone. Good morning, good noon. Lapit tayo mag-lunch. I hope nabusog tayo sa kaalaman at excitement this morning. Nabusog means filled, our hearts and our stomachs are filled with the amazing talks that we heard this morning and this community that we are building. It's so amazing to see so many people gather here virtually, 200 plus I see in Teams, and then many more as we keep growing this community of curious adventurers. Okay naman, I can now share my closing remarks. As we bring the seminar, The Search for Life Beyond Earth, to a close, it is worth reflecting on just how far our thinking has traveled today. Within just a few hours, we have moved from understanding the fundamentals of life to imagining its existence far beyond the planet we live in. And yeah, we all deserve a good lunch after this intergalactic and interplanetary traveling. And in the seminar, we began with the foundations of astrobiology, a field that dares to ask one of humanity's oldest and most profound questions: are we alone? As Miss Angie emphasized in her talk, what makes this question powerful is not only the possibility of an answer, but the journey it demands across scientific disciplines and across borders. And wherever phase in the journey you are so far, we are excited to invite you and to have you as part of this journey with us. We also explored different hypotheses on how life began on Earth, grounding our understanding of life as we know it. Professor Paola took us on to our neighboring planet, Mars, reminding us that the search for life beyond Earth must remain anchored in evidence, guided by experiments and sound scientific reasoning. From there, we ventured even farther to the icy moons of our solar system, and Miss Savannah showed us that we could take inspiration from natural biology of the cold environments here on Earth and make educated guesses on how life might flourish to the subsurface oceans of the similarly cold environments in Europa and Enceladus. And and in other moons. And last but not least, Bernard brought us behind the scenes of discovery, introducing the tools and methods that make this search possible. It is interesting to learn that fundamental concepts such as chirality or handedness of molecules can guide us in our search for life outside our planet. Everything is really connected, no? And beyond science, beyond the data and discoveries, something really important emerged today, no? Connection, yeah, to that point. Today, we brought together international experts. We're very grateful for their time and for sharing their knowledge and also their passion and showing you by example really life and career, fulfilling careers in the pursuit of this field and of science and of discovery for humankind. And also in this virtual room, a stadium, we have our students, young professionals, and curious folks from all around the country, maybe even the globe, who represent the next generation of our explorers. And what